All right, so we'll get started. My project topic was venom use, including injection in vertebrates. So, kind of the introduction and background. Couple questions I had when starting out this research project was, how is venom defined? How did venom evolve? And we'll look at some diversity of venom evolution. How do different organisms use venom? And then how is venom used in the medical field? So, overview of the presentation, we'll just kind of go in order and address those questions. So, first we'll start with what is venom, and then we'll go to venom and evolution, vertebrates and venom, and then venom and medicine. So, what is venom? It's defined as a diverse group of chemical components that disrupt some type of physiological functioning of the victim. Venom glands are usually modified salivary glands. And then an exception to this is the duck-billed platypus, which we're going to take a look at later in the presentation. Most abundant components of venom are peptides and proteins. So that is actually a difference that we're going to look at. So since stomach acids can denature and inactivate both peptides and proteins, it's generally harmless to swallow venom, but comparatively, it is harmful to swallow poisons, which are composed of other organic compounds such as alkaloids. We're going to take a look at that on the next slide. Okay, so we're going to take a look at venoms versus poisons versus toxins. So, venom are toxic secretions produced by specialized cells in an animal that is delivered to another organism via delivery mechanism to produce normal or disrupt normal physiological functioning. So, active versus passive use. Active use is more like predation. So, for example, like a snake going to capture prey. And then passive is more defensive. So, example, like stepping on a stonefish, they're not coming right at you. So, it's most often used to achieve rapid prey incapacitation, and it's produced endogenously. So, that's within the animal itself. So, poisons are bioactive compounds that disrupt the biological function of an organism upon passive administration without the involvement of a delivery system. So, usually delivered through inhalation, ingestion, or absorption through the skin. And then it's produced exogenously, so synthesized by an organism, then harvested by eating that organism. So, many poisons are derived from plants or microorganisms, and poisonous dart frogs actually produce their poison this way. And then toxins are any biodegradable compound of a natural origin that can disrupt biological functioning of the target, irrespective to the mode of delivery. So we'll go on. Evolutionary complexities of venom. So, venom has evolved independently on at least 30 occasions over multiple lineages. So, evolutionary factors. Diversity of venom is kind of dependent on various factors such as geographic location, diet, predatory pressure. And then recent studies suggest that venom evolution may have had a rapid initial diversification due to a new ecological niche, which is followed by a longer period of purifying selection. So, more effective venom, and then venom is developed to target a specific predator or prey. So, evolutionary convergence in composition and biochemical activity. So, composition being amino acids, peptides, proteins, carbohydrates, ions, and salts. Um, Various groups of animals have parallelly or convergently recruited identical venom components through various protein subfamilies. And although there's a diverse chemical composition, venoms will often target similar receptor types. So, diversity in venom delivery, there's fangs, beaks, spines, stingers, and harpoons. We'll take a look at some of those later in the presentation. And there are no vertebrate classes that are exclusively venomous, but almost all, Aves is the exception, have venomous representatives. And then the earliest venomous vertebrae, conodonts, so tiny fish-like marine vertebrae. All right, so now we're going to take a look at the diversity in venom use. So we'll start with fish, specifically chondrichthyes and osteichthyes. And then in the Scorpionidae family consists of well-known venomous fish such as scorpionfish, lionfish, and stonefish. So a little about each one of those. Um, Lionfish have long, thin grooved pelvic and anal spines that administer venom, and then no venom duct, and the integumentary sheath is covering the spine ruptures and then delivers the venom. For scorpion fish, they contain venom glands when they contain venom glands wherever another animal comes in contact with it. So the sheath retracts and then the mechanical pressure secretes venom through the spines. This is an involuntary act. And then for stingrays, the spine is covered with a layer of mucus, and venom is protected by an integumentary sheath covering the venom glands. And the stingray barb, which you can kind of see right back here, so that is made is this like a sharp spine made of cartilage that can pierce through the skin of another organism. Venom is then released, causing pain to the wound, and then venom contains cardiotoxin and respiratory depressants. So we're going to continue with the diversity in venom use. We're going to look at reptiles, specifically venomous snakes. So snake venom is most well studied. There are three main types of snake venom: cyclotoxins, poisonous substances that destroy body cells via necrosis. Neurotoxins are chemical substances that are poisonous to the nervous system. They disrupt neurotransmitters by blocking reception sites or reducing production. Hemotoxins are blood poisons that disrupt normal blood coagulation processes. Snake venom glands are usually situated on the head, so behind their eyes, with a duct that delivers venom to specialized fangs that are found in the mouth. So there is a compressor muscle towards the head of the snake that squeezes the gland that forces venom into these fangs. So different anatomical classifications of snake fangs too. So there are epistoglyphus. So these fangs are at the back of the mouth. Proteroglyphus fangs are in the front of the mouth. Solenoglyphus fangs are retractable and then hollow front fanged. And then aglyphus is without fangs. And snakes are estimated to envenomate over 1.8 million people each year. So just these anatomical classifications, you can see epistoglyphus right here, and then proteroglyphus, fangs at the front of the mouth, and then some of these can be retracted, and then aglyphus without fangs. So we'll just look into specifically the viper family. So viper venom is continuously produced and stored within venom glands. Vipers are solenoglyphous with large groove folded fangs and the maxillary tooth apparatus. They have a sit and wait predatory lifestyle. And then viper venoms are highly proteolytic, so they're able to break down proteins. And they contain proteases, esterases, a bunch of other chemical compounds that can lead to acute thrombosis. And then certain PLA2s found in viper venom can also result in skeletal muscle necrosis. All right, so we're going to continue to mammals and the duck-billed platypus. So mammals do not exhibit a large diversity of venomous organisms. However, there are some unique delivery mechanisms in mammals, which I think is worth looking at. So we're going to take a look at the duck-billed platypus, which are the only representative of monotremes to secrete venom. They have extra tarsal venomous spurs that are located on the back of the legs of the male duck-billed platypus, but not the female. You can kind of see them right here. All right, so these spurs are connected to venom-producing curl ducts, which are located on the upper thigh. This is known as the curl system. And then, unlike other vertebrate examples, the venom glands have originated from sweat glands. And although the gland development is different, the chemical components of these toxins are very similar to other venomous lineages. The venom contains also similar genes to like genetic organization and then peptide structures to snake and lizard venom. So the curl system is inactive during non-mating seasons, and then during mating seasons, these spurs are used as an offensive weapon to dominate over other males in mate selection. So we'll continue on to the next section, which is medical uses of venom. So anti-venom injections were first developed by Albert Calmette against the Indian cobra. So venoms are extracted from an organism, then administered into other animals such as sheep or rabbits at a non-lethal dose, which elicits an immune response. The immune response to the venom creates antibodies, and then those are purified for clinical use. So monovalent antivenom is effective against the egg, like a specific species of venom, and then polyvalent is effective against a range of venom-producing species. So snake milking. Milking snakes is done to extract venom that is used for medical purposes. Dangerous snakes are sometimes put under general anesthesia. Snake heads are grasped, and then snake jaws are opened to expose the fangs. Venom is then squeezed out and put into glass vessel. And there's a little demonstration.
Rightio, so I'm going to grab the snake out of the enclosure. I'll get you guys to jump back at that point because he's quite big and he'll swing around a fair bit if he wants to. We'll then come over here. I'll swing him onto the pinning pad and using the pinner. So nice and soft to protect the snake and protect myself. It's slightly tacky as well, so it gives me that split second of time where he slightly sticks to it, but it doesn't hurt him at all.
Yeah.
Then we're going to grab him and hopefully it all goes well.
We'll be quiet and we'll let you focus. Oh my goodness. That is a huge snake.
Yeah, you can see him a bit cranky. He's not just about this arrangement.
Okay. Where would you like me to go? Just
right. So you just hang just there.
Okay.
I'm going to swing him, and then I'll get you to come in and just grab this back end for me. Because once I get him by the head, he's not going to be happy.
Yeah, go ahead. Just focus. Do what you're doing. Grab the body.
Yep.
Got him. Oh my gosh, he's so powerful.
Come over here.
Yeah, that is a big nugget right there.
Right, yeah. So now the trick is, he's not a stupid animal. He wants to like me, not the jar. Take your finger as the target.
Oh, wow, that is a huge venom gland.
Give his venom glands a bit of a massage, try and get a little bit more. But yeah, you can see him just chewing on that thing.
Wow, look at those fangs. Massive fangs for an elapid. Wow. That is like a grappling hook of teeth. Can I move on just a second?
Yeah. Well, you can see him still chewing, still protecting venom.
Venom is still dripping.
And he's not letting go.
And the power in this snake's body is unbelievable. So big around, I can barely get my hands completely on it. That is a massive snake. I never imagined seeing a mogul of this size. So how do you get the snake off of the venom body?
A little bit of manipulation, or there we go.
Ooh, my goodness.
And what we're going to do is I'm going to get you to put it in.
Okay.
I promise I won't let go till I've
Body first?
Yeah, body first.
Here we go.
Right, yeah. Now jump on back, because this is the bit that goes a bit sketchy. Ready? 3, 2, 1.
Oh, nice job. That is a risky little game right there. Woo!
So that was kind of a cool video of a demonstration of milking the snake. So there are some problems with antivenom. So receiving antivenom can induce allergic reactions, or called antivenom sickness. Availability and affordability of antivenom poses an issue in developing nations. Another limitation of venom therapeutics is that there haven't been a wide range of studies on them. Studies are usually done out of necessity, medical significance, or ease of collection. So other medical uses of venom. So scorpion venom is currently being researched to prevent HIV. Venom of spiders and scorpions are both being researched as a better alternative to plant pest control. Research is being studied on bloodworms for their venom, as venom, a glycotoxin, causes a long, abundant yield release of a neurotransmitter acetylcholine into the neuromuscular junction, which results in muscular twitching. So this is useful for scientists because they are able to see how muscles and nerves interact. So conclusion, there's an interesting and diverse evolutionary history of venom development in vertebrates. Future research is definitely needed for a wider range of venomous vertebrates and understanding those in order to have a more complete understanding of venomous organisms. So as more venomous organisms are studied, then it may open avenues for more medical research in venom. Thank you. And then here are my references and the media references. Thank you.